This method employs topical application of nanoparticles to image microscopic tumours, especially in malignancies that do not yet have access to the vasculature, such as the metastasis of ovarian cancer. This technique is ratiometric, imaging targeted and non-targeted nanoparticles in a single scan to diminish false signals and reveal the true extent of the tumour.
These nanoparticles can be administered by intraperitoneal or intravenous injection for high-precision imaging of ovarian cancer and many other tumor types.
For gold nanostar core synthesis, place a conical flask containing 800 milliliters of freshly prepared ascorbic acid solution on a magnetic stir plate at 4 degrees Celsius and induce a steady vortex. Quickly add 8 milliliters of freshly prepared tetrachloroauric acid solution to the vortex. Within seconds, nanostars will form and the solution will assume a dark blue color. Pour the nanostar suspension into 50 milliliter conical tubes for centrifugation and aspirate all but the last 200 microliters of supernatant from each tube at the end of the spin. Use a micropipette to resuspend the nanoparticles in each tube before pooling all of the nanoparticles in a single dialysis cassette. Then dialyze the nanoparticles for at least 3 days against 2 liters of deionized water, changing the water daily. For silica shell formation, first add 10 milliliters of isopropanol, 500 microliters of TEOS, 200 microliters of deionized water, and 60 microliters of dye to a 50